Hi, everyone. We're excited to be back at the Learning Salon. Today, we are super excited to have Gina with us. Dr. Gina is a professor at UCLA and works on the how we can study the science of sleep. And in some of her talks, she adds dreaming, which is something that I would love to hear more about. I've seen a scientific kind of focus on journaling of dreams and how they relate to everyday life. And those are interesting, but it would be fun to see how they relate to the EEG and neuroscience and psychology of sleep. I want to remind everyone that the Learning Salon is a forum for discussing bridges and contentions between biological and artificial learning. Obviously, we are talking today focusing on biological learning, but we will also consider from some aspects what kind of things the notion of sleep can teach our models and computational approaches as well to learning. Just a reminder that there is, for some reason, a link that was emailed by Worldwide Neuro that's the wrong email, the wrong link to another meeting. I have left the correct link in that meeting. So if you hear somebody being confused, please let them know. They will come back here. We will fix this with Worldwide Neuro. With that said, John is going to be a little late. He's on a train in Europe somewhere, and he's going to reach land at some point. Before waiting too long for him, I just want to remind everyone that we have a policy here of being curious, asking questions, but also being respectful. Remember that no kind of hate speech or any disrespect is tolerated here. However, discussing, disagreeing in the service of understanding is completely okay. We understand that you might be from a different field. Please ask your clarification questions in the chat, and we try to address it during the talk. But your more elaborate questions, please put them in the Ask a Question area below and vote on each other's questions. At the end, we will call some folks on the screen to discuss with us. And without further ado, Gina, please take it away. Thank you so much for being with us.
Hey, this is my pleasure. Thank you so much for the invitation. It's great to be here. And I am definitely open to talking about dreams. I've been in the sleep field for 35 years now. But I've always studied sleep in regards to animals who you can't ask if they're dreaming. However, a lot of the brain activity we see going on during sleep very much could be dreaming. It's reiterating paths through mazes and that kind of thing. And certainly when you have an animal or a person with disinhibited muscles, normally when we dream, our muscles are actively inhibited, so we don't act them out. But there are certain conditions where that inhibition mechanism is gone. And so you do see people and animals acting out dreams. So they probably do dream, other animals besides humans. It's very, very likely, but again, we can't really ask them to do so. Anyway, given that, I'm happy to answer any questions about the dream state and perhaps a little bit about dreams themselves. There's a really great book that just came out about perhaps the function of dreams that I think is really well informed in the science. All right, so with that, I'm going to start my slides and give a brief talk. It's not supposed to be very long, so we have lots of time for discussion. All right, so let me share my screen with you and go back. Yeah. Can you see my screen? Did I hit the right buttons?
No? This time I can't.
You cannot. Okay, maybe I didn't quite hit the button thoroughly. Yes, it just asked me to allow and I didn't say. So now can you see it?
Yes.
Okay, great. Okay, whoop, I misspelled cognitive. But anyway, cognitive neuroscience is sleep. And then as a secondary thing that I'd like to just talk about is sleep really important for AI as well. So this is my laboratory funded by the National Institute of Mental Health. A lot of the lab members here in a Zoom picture. We've been on Zoom for about a year and a half now, but we're finally back in person, and I'm really glad to see that everybody hung out and stayed with us. So what can sleep do for AI? And I'm hoping we can address that a little bit because a lot of people here are interested in it. But does learning require sleep might be an important question for AI as well. As you can see, sleep is super important. It's homeostatically regulated, and we just can't get by without it at all. If we don't sleep, we die. So if we're sleep depriving ourselves, our bodies will force us to get it in whatever situation, even dangerous situations that it can. So every animal sleeps, from whales to worms, and even jellyfish, which don't even have a central nervous system. They also sleep. Here is an image of a jellyfish called Cassiopeia. And here it is awake, pulsing away. They are an upside down jellyfish, which is why it looks so funny. But even they sleep, and when they sleep, they just pulse a lot more slowly. If you deprive them of sleep by putting a jet of water or air on them, they will wake up and start pulsing more quickly. But if you do this again and again, their sleep is also homeostatically regulated. So it must be really, really essential for anything with the nervous system. And here's just how the jellyfish pulses during the day, and then it's slower pulsing at night. And there are periods of time which were not talked about in the paper that was published by Caltech 2 years ago. But there are even times when they're not pulsing at all. So like I was just talking about, when we have REM sleep, we have atonia. Our muscles are not active. We're not acting out our dreams. So it seems that every animal studied has perhaps even two states of sleep. One, which is called slow wave sleep, which perhaps in the Cassiopeia would be these slow pulses, and one which is REM sleep, which is paradoxical because our brains look like they're really active, but our muscles are inhibited. This is just roughly how sleep is organized across the night, across the 8 hours of night. And this is sleep in my sister-in-law, measured through her Fitbit, which isn't always the most accurate, but I thought that this was just very classical sleep structure, so I'm using it for an example. And the first state we go into after we fall asleep is N1 and N2 stage sleep, and then into our deep slow wave sleep if we're young and healthy. As we get older and older, we get less and less of this deep slow wave sleep where we have in our EEG, our electrical signals in our brain just shows big slow waves sweeping through. Then we go into that N2 stage again, which is the transition to REM sleep, where we have sleep spindles. They're 1.5 second long bursts of activity where our hippocampus, which is our learning and memory structure, is uniquely connected with our forebrain. And it's thought to be during this N2 stage of sleep, which we get a lot of throughout the night, that we're really transferring memories from the short-term to long-term structures and integrating them with each other in the cortex. And then lastly, we go into REM sleep, which has in our hippocampus, this learning and memory structure, a theta rhythm, which is about 5 to 10 hertz in frequency. And that's a rhythm that really seems to be able to coordinate the timing of incoming activity to strengthen some things and weaken other things. And that's this red state up here. We get that every once in a while as well. And it seems to occur right after we've transferred memories out of the hippocampus to the cortex. And so it might pose a very unique and essential function. If we don't get REM sleep, even if you get all the other states, you will also die in just as short a time, which is just a couple of weeks. So what are we doing when we sleep? Well, it seems like what our brain is doing, the analogy would be it's cleaning up after the waking party. During wakefulness, our brains are utilizing a lot of energy. We are getting a lot of new information in. And what we need to do when we go to sleep is get all the party guests out and clean up. We need to figure out, first of all, what to throw out, what things that the guests brought in that we don't want to retain forever. That's really important for not saturating our brain. And then we also need to figure out what we need to keep, what's important, what needs to be consolidated and integrated in with everything that we know. So part of throwing out the trash occurs in deep sleep. And we've only known this for the last, I don't know, 5 or 7 years, maybe a little longer than that, maybe 7 years, I'll say. But the brain's lymphatic system really seems to pump the garbage out. It might even be a function of those big slow waves that sweep through our brain to actually just take the garbage that has been like unwrapped presents, just proteins that are misfolded, and just sweep them out. And the reason why slow waves can do that is because the majority of our neurons are on and off at the same time at the peaks and troughs of these slow waves, which interestingly swells and then shrinks the membranes of our neurons synchronously, which then would squeeze the extracellular space and then relax it all synchronously. So it can work something like a pump system to pump out the garbage. So these are some of the papers that were published in this field, really showing us that if you inject tau proteins, for example, tau is one of those misfolded proteins that aggregates in Alzheimer's disease. It gets cleared out only during sleep, not during wakefulness, and not even during REM sleep. During that slow wave sleep is when that tau gets pumped out. Another thing that happens in slow wave sleep is we can build proteins. We can build new proteins that need to be built when we have learned something new and we want to convert it to long-term potentiation. So the proteins are built in sleep during sleep because growth hormone is released in a giant bolus. And if you inject like a radioactive amino acid, the only time when it really gets uptaken into proteins and built into proteins is during slow wave sleep. So slow wave sleep really seems to be important also for building proteins. Another thing that happens actually in the first 20 minutes, perhaps even of sleep, is that we're able to restock our energy stores. And that happens because as we're awake, the longer we're awake, the more ATP, which is the energy, it's like the fuel for the brain made by mitochondria, that gets broken down to ADP, then AMP, and then just adenosine. So adenosine builds and builds the longer we're awake. And when we go to sleep, within the first 20 minutes of sleep, our ATP levels are really brought way down because ATP is repackaged by our mitochondria into ATP. Adenosine is repackaged to ATP. And that's really also another important function of sleep. So, and then the last couple of things that I want to talk about is integrating new memories, new items that we've learned during the day in that N2 and REM sleep state, and consolidating memories and dreaming up new solutions in REM sleep. So one of these things that I talked about as occurring in N2 sleep is sleep spindles. You can see they last about a second or so. And when sleep spindles occur, you also get unique communication between the hippocampus and the prefrontal cortex. Also, what happens is something called the Locus Cyrillus, which means blue spot. It's deep in our brainstem. It is silent before each sleep spindle and then fires in a burst at the end of each sleep spindle. The Locus Cyrillus spikes after a period of silence. And what the Locus Cyrillus does is it helps write those memories into our cortex. So let's skip this for a second. I'll just show you what the Locus Cyrillus, here it is in the brainstem of a rat. We've got it as well. And the Locus Cyrillus projects all over the forebrain and also to the hippocampus here. And what it does is it helps learning, and it helps really rapid learning called long-term potentiation, helps strengthen synapses between neurons. And when it's not there, you can still get LTP, but it's a lot harder to do. And so when it is there, it's just really rapid. Okay, and when it's also not there, that's the only time you can actually get depotentiation, which is weakening of synapses, which is also paradoxically and a really important part of learning. When you learn something, you're not only learning what is there and what is, but you're also learning what is not. And that's an important thing. So sleep spindles incorporate new information into our existing neural circuits, and REM theta prunes those redundant connections. So, for example, if you learn something new, you need to incorporate that into the schema of everything that we know. And that happens through sleep spindles. That's when the hippocampus is corroborating and consolidating that memory out to the cortex. And then through REM sleep also, during that theta period, REM sleep is the only time, except for those few seconds before each sleep spindle, REM sleep is the time when the locus cyrillus is off and you can get depotentiation. And that happens in memories that are very familiar and have already been consolidated from the neocortex, from the hippocampus to the neocortex. And then it also happens for anything that seems to be irrelevant, things you don't want to remember long term. So it's a very synapse circuit specific erasure that happens during REM sleep in that theta state. And if either one of these goes wrong, if you don't have good sleep spindles or you don't have REM theta, that incorporation of that new information is flawed. It's not sewn into the schema. And if that REM theta doesn't happen well, for example, if your locus cyrillus is still active in REM sleep and providing norepinephrine, which is its neurotransmitter, to the forebrain, instead of erasing those things you want to erase that just get continually reinforced. And this seems to be the problem with those that have post-traumatic stress disorder, for example. They cannot learn a safety signal and incorporate that safety signal in and the safety context in with what was previously a fearful memory. And so instead that fearful memory just gets reinforced again during REM sleep. People with PTSD have a lot of nightmares. They have difficulty falling asleep because they don't want to fall asleep because it's a stressful time for them, the time they can't control what they're thinking and they can't dissociate. And it is also a time when there's a lot of evidence that the locus cyrillus providing norepinephrine to the forebrain does not turn off normally like it normally does. Another condition where people don't have good sleep spindles is schizophrenia. And that is another condition where memories might get mixed up with one another and associations are not schema are not well separated one from another. All right, so let's just go over quickly what we've talked about the function of sleep, which is to clear out the garbage, to restore energy, to build new things that we need to incorporate, strengthen new memories that have been made. And then during that transition to REM N2 state, we can integrate new things that we've learned in the hippocampus and consolidate them to the cortex. Something that we're going to talk about in just a minute is also how our cortex integrates the schema with each other's. So we transfer information and integrate it. And then during REM sleep, we talked about the lack of locus cyrillus activity, the lack of norepinephrine. You can actually clean out any kinds of memories and connections that you don't need any longer. For example, things that have already been consolidated to the cortex, you can clean them from the hippocampus. And you can also prune away anything that needs to be pruned away. That happens especially during development. And with all of the activity that happens in REM, immediate early genes, you can actually get learning all over again and sort of reimagine and have creative insights during that state. Okay, so I just wanted to just talk one more time before I get into one other thing, which is how the states of sleep are organized. And this might be also something to take into account if you're trying to design an AI system, which is that when you're awake, you're behaving, you're bringing in all kinds of new information. During slow wave sleep, when you're unconscious, you are cleaning things and sort of shoring up the important things. And then during REM sleep dreaming, you are actually consolidating, and the transition to REM, you're consolidating and then erasing things that are not important. And this happens in cycles because during REM sleep, you're actually learning new things, making new connections, which create new junk and utilize a lot of energy that needs to be restored in slow wave sleep, back and forth until the job's done and then you wake up. Okay, so the last point I want to make before we go, and I know we've already gone about I think about
Oh,
15 to
take as
17
much time
minutes.
as you want. Take as much time
Oh,
as you
good.
want. Usually people
Okay.
go, it's fine.
Okay, great, great. So these next two slides are my last two slides, but they're going to take a little time to go into. And this is how sleep serves to consolidate the things that we've learned on a very mechanistic level. So in our hippocampus, which is this structure that's really important for novel associative learning and memory, if we don't have a hippocampus bilaterally, we cannot form new associative memories. They are these cells called pyramidal cells, which are organized in a particular way. It has the cell body, it has proximal dendrites, and then it has way out here at the far away is called the distal dendrites. And the inputs to the hippocampus are different in these two compartments. You might want to call these two compartments. And the other thing that's different is that this theta rhythm that I talked about before is at opposite phases at the distal, at the proximal dendrites, then from the distal dendrites. So when you learn something new, it goes in and synapses at the locus cyrillus in our brainstem before it gets to the rest of our brain. And if it's a very strong new input, then it can activate our locus cyrillus and cause us to pay extra attention. The locus cyrillus and surrounding areas also activate the area that sets up theta in our hippocampus. And that information travels to the neocortex, which then travels through the trisynaptic circuit into our proximal dendrites here through what's called Schaefer collaterals, and causes long-term potentiation because you have lots of norepinephrine, which helps you immediately get long-term potentiation. The inputs coming at the peaks of theta when the postsynaptic cell is most able to respond, and you can get really quick Hebbian plasticity, long-term potentiation very easily here in these proximal dendrites. But this is called the novelty encoding circuitry. And one other thing that the locus cyrillus activation does is it inhibits these distal inputs. It inhibits inputs through what's called the familiarity encoding circuitry. So even if something is, even if the novel input is familiar to you, if it's a very strong and different kind of activation, you can get activation of the locus cyrillus, which inhibits that familiarity. You know, maybe I know this, but I've never heard it this loud before. Maybe it's something I should pay attention to. And it inhibits this circuitry. So this novelty encoding circuitry is free to do what it does, which is learn something new. Another thing that happens when we're awake and learning is serotonin systems in our brain stem are on. And serotonin, which is also called 5-HT, also prevents information from coming into the familiarity encoding circuit from reaching all the way up here and causing the cell to fire. It leaks out through a leak current and reduces the inputs through this what's called a temporomonic circuitry that comes in from another area of the cortex. So our brains are geared to learn something new. And because norepinephrine is present, what it does is it blocks depotentiation. We couldn't erase anything during waking, even if we wanted to. So the first stage of sleep that is important for learning memory is this N2 stage of sleep. And that's where these sleep spindles happen that I talked about before. And when sleep spindles occur, what they do is they arrive particularly strongly out here at the distal dendrites. During this stage of sleep, also serotonin is low and norepinephrine levels are low. So they are present, but they're much lower. And so we don't have as much leak current. And so these distal dendrites can actually create a dendritic spike, and you can get LTP. The third thing that happens during N2 sleep, which I haven't mentioned yet, are these Pgo waves. These are waves that come from an area just below the locus cyrillus, and they're big glutamatergic waves that also project all over the brain. And they seem to be probably the instigators of dreams. So these P waves cause massive depolarization, and they are targeted also at these distal dendrites. So it's different from wakefulness, but it can cause in and of themselves, because there's so much glutamate released, so much excitation, they can cause long-term potentiation at distal dendrites, not only in the hippocampus, but also out here in the neocortex. So when the cortex and hippocampus are uniquely connected during these sleep spindles, the hippocampus, which is activating these memories, can write those memories out here to the distal dendrites in the cortex. So also out here in the distal dendrites of the cortex are cortical-cortical connections, and that they can be strengthened during these Pgo waves and during these sleep spindles. So that's where new connections between things that you might not think are connected can really occur during this N2 stage of sleep, when so much activity is going on out here and a lot less is going on at the proximal dendrites. All right, finally in REM sleep, we have a novelty encoding circuit can be refreshed. And what I mean by that is that it's another time when activity out here in the distal dendrites is emphasized, and because you don't have any serotonin and no norepinephrine, what's previously potentiated out here can actually reach all the way to the axon hillock and cause this cell to fire. And it will be firing at a time when these inputs are coming in, but that's at a time when the proximal dendrites are at the farthest away from their action potential threshold. They actually are not coming in at the same time. And what happens when the cell is firing, these dendritic spikes are making the cell fire, but no inputs are coming in here in these proximal novelty encoding structures, then what you get is heterosynaptic depotentiation, which means that if this is the postsynaptic side firing and the presynaptic side is not knocking at the door, so it's kind of like ding dong ditch. After a while, these synapses are depotentiated, and they can only be depotentiated during REM sleep because there's no norepinephrine present, and norepinephrine protects anything, no matter what happens, it protects it from depotentiation. So that's when this novelty encoding circuit can be refreshed, when those synapses can be depotentiated, weakened again, so that they're fresh and ready for the next day of learning. So this is when we can clear away what's already been consolidated. And I just wanted to say one last thing here, which is the consolidation signal is intrinsic to the circuit itself. So I talked about the entorhinal cortex layer 3 coming in here. When the hippocampus consolidates memories out here to the cortex, the first area that it stops is the entorhinal cortex. And so consolidation here in layer 3 feeds back into a stronger signal here at the distal dendrites, which is the thing that causes heterosynaptic depotentiation in the novelty encoding structure. So this is the familiarity encoding structure, and just intrinsically weakens the novelty encoding structure. And that's how you can prevent a saturation of circuits and also make sure that everything is encoded efficiently. All right, so that's really the last thing I want to talk about. If you want to talk about dreams, we can talk about that, but I'll stop sharing my screen for now so I can see you all again. Let me go back to this and stop sharing my screen. There we go. Thanks, everybody.
Thank you so much. This was so fantastic. I wish I had taken a class.
Thank you.
I genuinely think I would definitely love to invite you for a longer talk at Microsoft Research, but I'm thinking there's so much here that AI and ML could learn from. And I'm familiar with the cognitive neuroscience of sleep in humans and fMRI and EEG. I didn't entirely know everything that you mentioned, and I learned so much. I really appreciate it. So you were one of our few guests who really kept the time.
Wow. It's good to hear.
Usually we say 20 and people go on to 40. So I really want to thank you for that.
Sure.
That being said, I have so many questions. There are already some questions posted. People are thanking you for in the chat. I just wanted to maybe start by asking a few questions. And I guess
I just want to say hello, Gina.
Hello.
And apologize. I raced from German Switzerland to French Switzerland to get into my hotel room.
Wow.
But I did catch the end of your talk, and I have questions too, but they're
Okay.
Why don't you go first?
Hopefully simplistic ones compared to what Ida will ask, I'm sure.
John, why don't you go first?
Yeah, I wanted to, we don't get to have a sleep expert on very often, and I'll be very honest with you. I'm a neurologist, and I've been interested in sleep glancingly, but it's very much in my peripheral vision. And I studied consolidation overnight versus during the day in motor learning, and I'm very interested in all that. But I wanted to take sort of a step back. I remember I visited, I'm trying to remember where it was, UT Southwest, and I met a scientist there whose name you will know, I'm forgetting, who did all this wonderful work on circadian rhythms and cell cycles and things like that. Mm-hmm. And so I have a real chicken and egg problem when it comes to sleep. Sure. On the one hand, I hear that, there's been entrainment in almost all animals to the circadian cycle, and that's basically sort of the origins of sleep. And then you have sort of clock cells, and they entrain to a more central hypothalamic system, and there's all that, the circadian world, right? And I don't assume to know how low down that goes, but I know invertebrates have sleep and stuff like that. Yeah. And then you have the kind of story you told, which is very much about hippocampus and cortex, and specializations which are very much mapping onto those structures. So I end up having a kind of cognitive dissonance, which is, on the one hand, you can have this circadian view that goes well beyond cortex and hippocampus. Yet then
plants even have a circadian cycle.
Right. So so what I meant by chicken and egg is, what I meant is what you're talking about, and this very interesting relationship between REM and the other stages of sleep and awake, and this very intricate dance between behaving and then consolidating and pruning and de-junking and all that. Is that a adaptation of this special cortical hippocampal architecture to circadian rhythms? So in other words, sleep came first.
I understand.
Then, do you know what I mean?
Yes.
Which round is it?
Yeah. Okay. So the way I think of it is that sleep, the functions of sleep take advantage of the circadian off time. And so the various functions, the more complicated we are, perhaps, the more functions there are to sleep. But even in C. elegans, the roundworm, there are two stages. Let me put it this way. Sleep is really important for synaptic pruning, the part that I talked about, even in C. elegans. And so when C. elegans develop, if they don't get their torpor state, they call it, but to me it's sleep. If they don't get that state, they will die, and they don't develop normally, and they die. So even in this very low, and then I showed you jellyfish. It looks like there's a question in the chat about how we know jellyfish sleep. Well, one of the ways we know that C. elegans, fruit flies, and jellyfish are really sleeping is because it is homeostatically regulated as well. Maybe there are many who wouldn't want to call it sleep, but if you define sleep as in the ways that we can define it for every animal, which is increased threshold to arousal. So it's harder to get a jellyfish to respond to you during this state. And then if you do, it's homeostatically regulated. So if you deprive them of this slower state, they get more of it when they get a chance to. Let's see, how else do you define sleep? Let's see, homeostatically regulated, increased, there's a position usually associated with that. Even fruit flies have a sleeping position. C. elegans, it's harder to tell, they just stop moving. And then there's a fourth one, which I'm forgetting right now. But anyway, there are sort of, this isn't necessarily behavioral criteria, but if you give C. elegans or fruit flies or jellyfish stimulants that deprive us of sleep, like caffeine, they will also be deprived of sleep and will be more active, and then we'll homeostatically get it later. So So the idea of sleep for synaptic pruning, anyway, seems to be across all species that we've studied, and I would love to see species that are even more basic than the ones that I've presented today. But again, even those with distributed nervous systems and things that we cannot see, sleep spindles, and they don't have a hippocampus per se, they're still doing this, and it's still essential for life for them.
So that's really interesting. So could I be outrageous and say, okay, you just came up with these really interesting, almost core building blocks of what sleep is, right? And as you said, you hope that you could probably find them across the animal kingdom, right? And would it be allowed? I mean, I can't believe I'm saying this, but it's late and I had a long drive. But that it seems like any living system has to be on sometimes and off other times, right? And that on-offness, awake, sleep, maps onto almost every biological structure in some way. And when the structures get more complex, as you said, and you get up into hippocampal cortical relations, there's a particular flavor that core mapping takes on those particular biological structures. But you can see them have their incarnation in their own way across biological structures and across species. And you're basically just talking about the cortical hippocampal incarnation in those structures of this essential on-offness. Is that outrageous, what I'm saying, or does it kind of...
Oh, no, I don't think it's outrageous at all. I think you've got it right. Because that on-offness is really important for organizing things. For example, when proteins are synthesized, it's not just a simple, all the amino acids are in a milieu and they all just get together. There's machinery that's necessary for it. There's enzymes that need to be made in order to build certain proteins or tear them down. And all of that machinery needs to be built and available in a specific order that is organized, I think, through the circadian system. So if you don't have a good circadian rhythm, you don't have things available when they need to be available, and you don't have the enzymes there or broken down when they need to be there or broken down.
Yeah, it almost begins to feel almost like a new sort of almost thermodynamic principle of a living system, right? And it's interesting, like in neuroscience, we have lots of these sort of dichotomies, right? The learning phase, the maintenance phase, the exploration phase, the exploitation phase. There seems to be this sort of interesting, I wouldn't know if it's on-offness, but a kind of system has like two settings, right? Where it has rest and fix versus act. And it almost as though sleep is almost the universal dichotomous principle for a living system that then simply got more and more elaborate as the system itself complexified.
I would say, though, it's not exactly off. It's not exactly
No, it's not.
You're right. That's why I apologize for saying on-off.
Maybe you want to say offline, because
offline, online. Something like that. Exactly. Sort of, and we see that in learning. It seems like it's a very deep principle. Maybe for AI, right? If some people believe, right, that you have to sort of simulate life, not just intelligence. And I've always been skeptical of that idea. But maybe the way to actually not be is to say that if you really want to get intelligence right, you're going to have to, as Ida said, have offline, online. And offline and online is a property of living things. And can you have intelligence in a non-living thing that doesn't need to have offline and online versions of itself? It'd be interesting to... It's a deep issue, right? Do you have an opinion about that?
Sorry, before that, can I add one thing, though? The term offline replay and offline processing, online processing, however, came from reinforcement learning and computer science back into cognitive neuroscience in the early 90s. So I would just say it's a property of also, computational systems, the offline, online in our algorithms. However, what's missing was something that Gina said earlier in response to your question, which is homeostasis. These things are not used for homeostasis of memories or weight kind of appropriately. We are working on some algorithms, some early versions that we want to include homeostasis into learning. But I think it was very important that you said that earlier in response to John, that is indeed one of the functions. And that is one of the reasons the animal might die or might not be fresh to learn new things the next day. Sorry. So with that, please go ahead.
It's wonderful. It's wonderful. I mean, it's helping me really realize that there's something fundamental. And, to Ida's point, it's interesting, and then you go back to the start of the 20th century with Ebbinghaus, with this notion of savings and the idea that somehow it was offline. So it's, but it seems like it's a, do you think that you can't have a living system that isn't responsive to circadian rhythms? Would you agree with that? Is there any living system that you'd call alive that is non-circadian?
Well, not that I know of, but no, I would say no. But you can have a living system whose circadian rhythms are misaligned and not functioning well. That happens to us when we do shift work. But a lot of things start to go wrong. It's not that we die. We don't die within 2 weeks or anything like that, like we do with sleep. But we're much more prone to cancer. Our learning and memory systems are dysfunctional. And I do wanted to say something that Melanie, maybe it was Melanie Mitchell, someone in the chat said that she didn't understand what offline and online really means. And so I wanted to just mention what I mean by offline is that it's not taking in information from the outside anymore. Our systems are internally responsive and quite active, but we're not taking in, the party guests are gone. We are no longer unwrapping new presents brought by new party guests, and instead we have cleaning crews and redecorators, designers putting things in their right place and making things efficient. But the party's over. That's what I like to think. If the party doesn't ever get over, that's a sleep deprivation. The party guests stay and stay and stay. Then your cleaning crews can't do what they need to do. They can't. They can't sweep out all the big junk because they'd have to sweep around the party guests. And again, I'm getting the analogy and the physiology mixed up a little bit, but think about those big sweeping slow waves. You actually need silence, synchronous silence in large swaths of your brain in order to do that cleaning process. And again, you're not going to die immediately if you don't get those slow waves. And like I said before, the older we get, the fewer slow waves we have, but also the more things are going wrong in our brains, right? So it's a matter of efficiency in a way, yeah.
My last question, just so I understand, I'm just trying to, so it seems as though, given that there are precedents for this offline, online-ishness and arguments for it, do you think, when if plants have circadian rhythms, they don't have synapses, right? So again, do you think that the need for rest versus action is always going to be there for any system that behaves and learns and remembers? And so the circadian rhythm that might be present in plants, for example, was the way that you decided to carve up the rest period versus the on period. In other words, you took advantage of that way of carving up the day. It wasn't circadian rhythms and their mechanisms per se that are important for sleep. Sleep is a need of the kind that you talked about in homeostasis. And you co-opted the circadian rhythm to carve up the need for rest and on periods. Do you see what I'm saying?
Yeah, that could very well be. Yeah.
The origin of sleep, it was used by sleep. Do you know which one it might be?
Yeah, so some of the things that are regulated by the circadian system, the suprachiasmatic nucleus, one of the things that it instigates is the release of melatonin, and melatonin has a lot of activity, a lot of effectors. And it certainly affects the ability to learn. When melatonin is present, we don't learn very well. LTP cannot happen. So it's a complex question, and I don't think we could answer here, what came first, chicken or the egg, but it does seem beautifully integrated the way that it happens. Yeah.
Yeah. Thank you so much.
Yeah. Thank you, John.
Thank you so much. This is such a wonderful conversation. I find it also not just informative, but also inspiring, even though I've read so much about memory and sleep. So Mihai asked something very interesting. So really quick, he asks, do deep sea creatures have a circadian rhythm?
Oh, that's a great question. And I do not know the answer to that at all. Certainly, yeah, I mean, they don't see the sun rise and set. They probably don't get much temperature change associated with the sunrise and sunset down there deep in the sea. I don't really know the answer to that. A lot of deep sea and cave creatures don't have eyes, so they also can't be responsive to light in that way. Excellent question, and that would be essential, I think, to ask and to answer in order to figure out how universal circadian rhythms really are. Certainly, even deep in the sea, you would get monthly rhythms with the moon gravitational pull. So,
I love that.
yeah, yeah. So when the sun and the moon are both aligned, that would be a big pull in gravity, and when they're opposite sides of the Earth, we'd also get high tides. That's when high tides happen.
Very interesting. Actually, I will definitely go and read into that because, that's very interesting. I never.
I'm writing that down too.
Yeah,
great question.
So I want to ask you something related to that. So there were these studies in the, I think, 60s where they put some people in completely, dark, simulated caves. And then they observed how over weeks their circadian rhythm changed. And then their circadian rhythm turned out to be, instead of 24 hours, like 36 hours or some. Yeah,
not that far, but like 24 and a half hours. Some people's intrinsic rhythm is, 23 and a half hours, or 24 and a half, 20. I don't know if anybody's even gone to 25, but yeah, people have different tau that are intrinsic to their SCN, which is just reset by light to align us with the world around us. But we do have an intrinsic rhythm, yes.
Is it intrinsic or is it connected to the moon? So I wonder whether astronauts in space are going to have a different cave effect.
Well, I don't know about astronauts and their circadian cycles. I don't know. I do know that temperature has been measured in astronauts. And certainly they still need sleep, and they don't get a dissociated, naps all day long. It's consolidated sleep, but it's much shorter, probably because they're so overworked in their space. At least on the space shuttle, they had tons of work to do, and it was, it's a highly stressful environment. So their sleep averaged about 5.5 hours per night, but that's still a decent amount. Good question.
The main questions that I wanted to ask you were about the time when you were mentioning when norepi is not present, then there is the possibility of getting rid of some things that have already been consolidated, but not during wake. A lot of forgetting, however, happens during wake, including retrieval-induced forgetting and things that are based on something's just competing with one another in the field of memory. And also when we are surprised, or when certain kinds of things change in the world that requires us to change the focus of what we were doing and just search for another task set, either for survival or just to make sure everything is safe, then norepi actually is involved in signaling that and everything. So I wonder, if it's just norepi based, then there should be phases during sleep, especially when things in the world change rapidly, that would assist the dumping of some things that are kind of not super important right now to focus on something else. And also some consolidation does happen during wake, right? So I wanted to ask you those two things: forgetting and consolidation during wake.
Sure. So that's a multi-part question. We do know that norepinephrine is essential, or not essential for, but helps long-term potentiation. Too much of it floods our receptors and shuts itself off, but also causes panic, our attention mechanisms aren't working well. There's a competition between acetylcholine, which is an attention-related neurotransmitter, and norepinephrine. And so too much norepinephrine reduces acetylcholine and reduces our ability to learn. So if we're truly panicked, we also cannot learn. Our forebrain shuts off when we have way too much norepinephrine, which happens when we're panicking. So the ability to learn is much reduced. So there's a sweet spot right in the middle. Too little and we're not paying attention and we're too relaxed, and too much and we're panicking. But there's a pretty wide, thankfully, sweet spot in the middle. So you're talking about competing and so forgetting in other ways, not just erasing synapses, but forgetting by competition or interference. And yeah, I think it's really not well understood what happens. But if you've got something in working memory, and the hippocampus, working memory being in the cortex, it's trying to instruct the hippocampus, but it hasn't done so yet. And something distracts you and turns that searchlight of attention to something else, working memory just fades away, within 2 seconds. It's gone. And so if your cortex has not yet fully informed the hippocampus that this is important, you won't remember that thing. You know, if someone tells you their name at a cocktail party, and then something else distracts you, another even internal thought distracts you too quickly, you will never have written down that association in your hippocampus, and it is gone. You won't remember it. Period. End of story. It's just not even there. The trace isn't even there.
But I would say that was, it was retrieval-induced forgetting, which is you have learned some associations, but then let's say I have associated tree with oak and with pine. Someone keeps saying tree, oak, tree, oak, tree, oak. And then this kind of weakens the connection between pine
trees and pines.
Exactly. So that's called retrieval-induced forgetting that we have studied. Ken Norman has studied a number of people. We see it also in conversation with Aline Coleman. We showed that in networks of people talking to each other, also this happens. So if as a group we start to talk about certain things, but not other things that are closely related, we weaken the importance of them and strengthen these ones, for instance.
Yeah. So that's okay. So that brings up another important thing, which is people with Alzheimer's disease. One of the first things to be degraded is the Locus Cyrillus, which is the thing that provides norepinephrine. And so if the Locus Cyrillus is not functioning well, you actually cannot reconsolidate those things that you knew before. So whenever you bring something up, first of all, as you know, every neuron is connected to a whole lot of other ones, right? And not just encodes one memory in the hippocampus or even the neocortex, but is involved in many different memory networks that share some overlap, but also, you know, are not the same, right? So when a memory network is activated, all those other memories that any one neuron in that memory network are connected to could be heterosynaptically depotentiated. So you have the danger of erasing everything else but that one memory. And the thing that protects that from happening is norepinephrine. It protects that heterosynaptic depotentiation. But during slow wave sleep, norepinephrine is much lower. And it's really cool because at the peaks of every slow wave, all these neurons are active at the same time. Not all of them. There are particular memories that are being reactivated in particular orders and at particular times. And what norepinephrine, the Locus Cyrillus, does is it fires in a burst to allow norepinephrine to be at the synapses at the peak of each slow wave. And so I think that's really important for protecting all of those orthogonally encoded memories from being erased. If that doesn't happen, for example, in Alzheimer's disease, when your Locus Cyrillus is already compromised, every time you recall something, you are in danger of erasing a lot of other memories if you don't have norepinephrine and your Locus Cyrillus working properly at the proper time. Yeah.
That's fascinating. Related to that, and one last thing before we go to Melanie. Hi, Melanie. Good to have you here.
Hi, Melanie.
I wanted to ask, you had a slide that you mentioned, retouched briefly on PTSD. And PTSD is a field of basically both behavioral, neural, and computational study that I'm very interested in. And in terms of building models of it, we do think about, for instance, replay of the negative event, kind of maybe making associations to safe memories, because you replay so much the unsafe memory, but you also don't want to re-experience the body, so you kind of shut it down. So sometimes your body experiences it, but you don't consciously realize that's the reason, or you don't allow yourself for this memory to come up. So it seems like there is a direct path from that memory to the body that doesn't raise to awareness, which is a little similar to, you know, people getting into a certain body state, or dogs during sleep, which is the dog starts running, or the child it thinks it's eating, or you know, those kinds of things. I wonder what do you think is in common between maybe sleep and those kinds of states in PTSD where the body seems to remember, but we don't consciously remember what's happening.
Yeah, well, you know, the brain isn't ever alone. It's very intimately connected with our bodies, and there are descending projections of, for example, the locus cyrillus, the amygdala projects to the brainstem, and brainstem we don't generally think of as being a conscious, you know, area. So consciousness is thought to be in the neocortex, or associated structures, maybe even the hippocampus, but not in the brainstem. So when our bodies are responding, it's very possible that our midbrain and brainstem are quite aware of the threat and responding to what it considers to be a threat without much conscious recognition or processing of it at all. Yeah, I know we could go much further into this because it's really, really interesting. But it doesn't mean we're healing ourselves through that process. And I know you didn't mean to intimate that, but yeah.
I just wanted to say, you know, about the type of retrieval-induced forgetting and interference that I was talking about. You know, we were involved for a long time in debates in the sort of motor learning literature about this.
Motor learning of what?
Just in general, when you got interference.
Okay.
Now, there's a lot of people thought interference was about erasing of the kind you were talking about. And then I think we showed quite convincingly that wasn't what was going on. It was actually a more top-down, cognitively induced mistake. Than the fact that it was some erasure at the low level. So I think that, you know, the kind of, you know, word-paired associative learning where you get this retrieval-induced forgetting that has been studied, you can actually show that you didn't actually forget the thing that was interfered with, just because you can do other experiments to show that it's there. Actually, it's for some reason, for more top-down based reasons, you don't remember it because and
sorry,
and we've shown. So in other words, it seems like there's a way that you can get interference, consolidation, failure of consolidation, erasure, which is more of the kind you're talking about, and then there's another version. I don't think we quite know how that works, at least, but it does happen. But I don't think you have to talk about actually erasing or failing to form synapses or anything like that.
Yeah, so that is the reason I asked that. And as someone who has made models of that, John, and has done studies on retrieval-induced forgetting, what we think happens is that the weights of the connection becomes weakened, not that they become erased. And that's something that's in fact important, because there were different ideas in the memory literature. One was that you cut the connection entirely. The other one was that you just keep activating some more, so the weights just increase. So relatively, the ones that didn't increase enough will stay low, and this could be like, you know, that's the reason the disparity happens. Another one suggests you can weaken and you can strengthen. I'm of the third category in terms of the memory literature. Ken Norman is in that field. A lot of other people in the computational memory field are in that camp. And so what we think happens is that some memory just gets weakened its association. So imagine a subway, and you make visits to that subway so irregular that people forget that subway station exists. That's basically what it is. The subway station exists somewhere, but because there are no trains going there, meaning it's not being retrieved or accessed from other memories that are nearby, people forget that subway station exists. That's the meaning of forgetting that we are using in that field. And I'm making the subway kind of analogy because it is indeed a graph theoretic based kind of explanation of what goes on. And we've done models. Ken Norman has this huge 60-page paper in 2005. We have some newer models. There are many kind of like, there's a lot of work on that. I do distinguish that from the synaptic changes, which is why I was asking about what happens during wake when we forget something due to interference as opposed to that. And I want to highlight that this is not similar to motor cortex stuff because this is happening, we think, hippocampally. So I think there, and then the effect that it might have on the cortex, it could be long-term.
I mean, I agree with you. I don't actually think that, I think to your point, a lot of these sort of top-down interference effects, which are like word pair associative learning and retrieval-induced forgetting, it's much more often happening in the motor literature. And to your point, there was a belief, oh, well, it's motor, therefore it's got to be more of the lower level synaptic version of the story. Mm-hmm. And it turned out to be much more like word paired associative learning. And it was misleading because it was motor that it would be low level. It turned out to be much more of the kind that I was talking about. Actually, it was this different version. And
so there is actually an argument in the field, which I have a very strong opinion about, whether word pair associative learning is learning or is it recalling? Because if you already have the word pair associated, well associated from the time you were, 3 years old or younger, then instead it is an implicit reminder rather than new learning. And one might say some of the things you were talking about, Ida, which might be if you recall one pair of words and not another pair of words, it might be instead of erasing those other things, you're actually just inhibiting them. It's just through a close association, you're inhibiting the other association rather than weakening the memory. So I just want to bring up three possibilities, which is one is erasure, which you talked about before. Another is active inhibition. And then a third possibility is something in between. So depotentiation is actually the first step toward erasure, and during development and the critical periods, depending on whether the synapse has been activated or not, if it has been activated, you first have to depotentiate, then you do deeper than depotentiation, long-term depression, and then you actually retract synapses. Then you actually do a physical disconnection. But even in hibernation, when you've got a retraction of 90% of the synapses in your cortex, there remains a tunnel there. The extracellular matrix remains in place so that when you wake up, you can just reform the same synapses in the same places. Now, nobody knows if that happens because no one has ever marked these tunnels during hibernation, but it's quite a big possibility that that might be one of the ways that even despite hibernation, animals can remember where they stored their nuts before hibernation, even with 90% retraction of that. Yeah,
that's wonderful. I just added a link to a paper in which we kind of showed, we call it sample spreading activation and memory plasticity model.
Wonderful. I'm going to read that.
I would disagree that it's just recall, and here's the reason and why there is also learning. So we didn't, so in the lab, people learn to associate certain words with each other, right? This is not, the world doesn't associate words in this particular.
So they're wrong, they're words that they hadn't ever heard before?
No, they have heard them. They haven't associated them in this way. So it's the graph, it's the association.
So it's a novel association. Yeah. So that's not what I was talking about.
Yeah. So we do it for both. So in order to look for words that their association already exists, in the paper that you see there, we use words from, we use a NLP model on the Google corpus, and then get a co-occurrence matrix of words that appear together very often. So in that one, we actually look at that. And then we still show that computationally, if you activate one word very often, you can reduce the accessibility of a word that's closely associated with it. And actually, In one of them, we even say we do something that I call forgetting attacks, which is we want to decrease the accessibility of one word. In this case, we were trying to be funny. It was 2019. I don't know if everybody remembers what it was like. So the word was Republican, and then we would use Monte Carlo sampling to figure out what words needed to be repeated a lot so that the word Republican would be less accessible. So meaning the weights to it would be less in that graph. And what we found was something that I call it the kind of, or inspired by some work by other people in the field, we call it the amnesic shadow, is that when you do this, you accidentally, without wanting, diminish the weights of some other words too. And the word that we had accidentally diminished was truth, which was very interesting. So in that model, we allow for these kinds of plasticity. And in fact, I think one of the things that Ken Norman showed since the 2005 paper in Psych Review was that inhibition could really help. So I'm really with you that there's something in between, and I'm with you that we don't have a complete answer to that. And so, yes, our camp is closer to yours, saying, yes, some inhibition would happen too, depending. And it doesn't necessarily get consolidated. So it could be that during the experiment, I managed to make you not access the word Republican, but then you go out to the world, you have like, you know, you sleep overnight, the next day you're the same person. Maybe that's how a repeated advertisement or propaganda or such works, because it repeats so much that it gets consolidated after a while.
No, it's really scary to think that. But one thing that you had said there, I wanted to bring up a really cool experiment. And I don't know if any of you were involved in this experiment, but what they did is they took a bicycle and they reversed the handlebars so that so when you steered right, the bicycle went left, and then the opposite way. And they took adults who were bike riders and knew already well how to ride a bike and was already well consolidated. The schema was already well consolidated. They gave them three 15-minute sessions on this reversed handlebar bike. And they tested them if they could get a certain distance in a straight line, then then they passed the test. And then they had them nap. And those that had the most healthy naps, the naps with the most rich sleep spindles, something I didn't say anything about sleep spindles, but the more dense your sleep spindles are, the higher your IQ scores, and like 3 different types of tests. And also your ability to increase the density of your sleep spindles at night correlates with your ability to consolidate that memory. Anyway, those that with these shorter naps had the most sleep spindles were the ones that erased that erroneous, weird bike riding session fastest. They didn't tell these people, but they tested them again on this reversed bicycle after the nap. And those that had the healthiest sleep were the ones that did the worst. The ones that had the most disrupted sleep, the least sleep spindles, they were the ones that had retained that erroneous learning fastest. Now, this brings up a whole series of questions like, how does the brain know what to erase? I mean, you do want to erase something weird like that so you can ride your bike normally again and not crash, right? But how does the brain know what to erase and what to keep? How does it tag the things that you want to consolidate and knows the things you want to throw out? That's just
I'm absolutely fascinated by that experiment, and I have all sorts of opinions about which I will certainly not But could you just give the reference I did not know that a nap version of that experiment with the reversed handlebars had been done. So I would love to know what that was.
It was done by a group in Switzerland, so right where you are.
So that's really interesting. We'll find it, but thank you for that.
I will try and find it and send it to you, okay?
Just before we go to Melanie, I just want to say that's you raised.
Somebody put it in the chat.
Oh, perfect. So there's like a what I put there is just the YouTube.
Okay.
YouTube of how they do. It's not the reference.
Okay, good. Okay,
so
then we'll find the paper.
What I wanted to say is in reinforcement learning and memory, when we are building memory trees or models of memory, the question of what do you choose to erase has become super important, and it's not at all trivial, because you optimize different kinds of things. You optimize some kind of forgetting, and you get basically something that doesn't really need memory because it's basically like averaging over something, but you don't erase enough, then memory becomes useless because search becomes, linear, and it's
everything brings up, and
it becomes too slow to find anything. So it's a very fascinating question, and I think that we can learn a lot from that field. And I just wanted to say that I'm really fascinated that you brought up an example where, counter to usual examples, nap is impairing the new learning. And I think this is part of the reason that when some people go and have the first night after a sort of a PTSD, like traumatic event, usually hospitals make them not sleep the first night. And part of the reason is not to, I don't know, they think they don't want some of the memories to get sort of too long term or something. I'm not sure entirely the reasoning behind that, but.
Yeah, I can tell you what my model would say about that. And I think it is a good idea not to sleep right after a trauma. And in fact, you want to delay sleep until such time as you have calmed the locus cyrillus, which is that, alerting, alarming system. So just recalling, for example, you might want to send in counselors after a big traumatic event and the counselors might want to interview those that went through it and have them recall it. It's not necessarily a good idea to recall it unless you can, through that recall process, come to some sort of peaceful resolution that allows you to calm down. And for example, if you can realize in a way in the future you can get out of it and not have to go through that again, that feeds back, that learning feeds back onto the locus cyrillus and actually inhibits its activity. Because if you go to sleep with your locus cyrillus still active, with your stress system still active, then it's still active during REM sleep, then you have REM sleep with norepinephrine present, which just serves to reinforce things, not allow you to step anything down. So even after a stressor where you don't have any control, there's nothing to be learned necessarily about it, your locus cyrillus will, after 6 or 7 hours, calm down usually, unless you keep that stressor up. If it's still happening, of course, it's not going to calm down. But if you leave it in the past and after 6 or 7 hours, the locus cyrillus will calm down. So I do recommend sleep depriving yourself after a trauma for a period of time, or unless you can figure out a way to come to peace before. And one way people come to peace is by reconnecting with their worldview. If you believe that everything's going to work out in the end, even if this sucked right now, this was awful, in the end, justice will be served. That might be a way to reconnect. Prayer or meditation or doing something calming might be the thing to do. Yeah.
Thank you so much, Melanie. So sorry to keep you waiting this long. Melanie.
Hi, Melanie.
Hi. Yeah, no problem. This has been fascinating. Thank you so much for the talk. So I had a bunch of questions, but I'll just ask the one that hasn't been answered yet, I think. So you talked early on about the slow wave sleep and how that kind of diminishes in age. And that's really fascinating. And I wonder what's known about that, and also kind of the connection to learning, because we have different learning styles and abilities at different ages. So which way does the causality go there?
It's a fantastic question. And the great thing about aging is that it becomes so heterogeneous. Some people preserve their slow waves well into their 90s, and others it's gone by age 50. So it's very heterogeneous. But we do know, for example, in general, older people, it's more difficult to reverse bias. For example, their biases become more ingrained, if anything, and not less ingrained. So the loss of the synaptic plasticity in slow wave, with aging, might be related to the changes in sleep. And very much another thing that goes up with aging is insomnia and interrupted sleep. And if your locus cyrillus is starting to become impaired, then insomnia, first it's hyperactivity and then it's silence. So with the hyperactivity, insomnia would definitely be a function of the hyperactive locus cyrillus. So I think finding ways to preserve good, deep, slow wave sleep could be a priority for good healthy cognitive aging. So, and one way you can do that, one way to help preserve slow waves is to exercise during the day. Exercise is good for so many things. It's also good for sleep. It really keeps your slow wave sleep nice and juicy. I'm not going to say it.
So just to follow up, so somebody else asked this same question in the question list. Is it possible that maybe the causality goes in the other direction for that, as we age, perhaps, evolutionarily, we don't need to learn as much at age 50 as we did at age 4. And therefore the body just says, okay, I don't really need to have that kind of sleep anymore. So it's not that the sleep prevents me from learning. It's that my learning style now just says, I don't need that kind of sleep.
Yeah, no, I think you've brought up a great point, and that's also true. If we're not learning anything, we don't have as much REM sleep, or that N2 transition to REM sleep state, or the density of sleep spindles. And so in a way, it's responsiveness to our learning demands that makes it's a homeostatic response. Our REM sleep goes up, our transition to REM sleep goes up if we're trying to consolidate something new. It's, I guess, in the case when you're trying to learn something new and you don't have the machinery in place to increase those stages of sleep, that learning becomes and consolidation becomes impaired. Yeah, I think that's a great point. I would love personally to be able to learn like a 4-year-old does and to have less biases. 4-year-olds have lots less biases, but in a way our biases make us quick thinkers, which could also lead us down the wrong path. So
yeah, I mean, Alison Gopnik, I don't know if you know her work. She has a fantastic set of papers on like why we have these different learning styles from babyhood up through, grandparenthood. And it makes sense.
Yeah.
But thank you.
Yeah, that's a great question.
So we have a couple of other questions. Samantha asked me to ask for her, but Melanie, please stay up because usually you always add something very.
Yeah, please.
Samantha Walters asks, and also whoever wants to join the screen, please write in the chat. I don't want to call people out, but there's a lot of questions. So it would be great if some of you can ask your questions also. I wonder, Samantha asks, I wonder what features of sleep in relation to learning and forgetting would be most important for implementing in artificial learning systems?
Yeah, that's a good one. Well, as you have, one thing that might not be so important in an artificial learning system is energy restoration, right? I mean, if you're always connected to the electricity, your energy is plenty. So, okay, so, and also maybe growth isn't really, you don't have growth hormone in a computer, right? You don't need that. So what is it about slow wave sleep that might be still implemented in AI? One of the things that happens during slow wave sleep is that protein synthesis helps stabilize what you've learned, so it's not all erased. So if that's also not a problem with AI, it might be that you can dispense with that state of sleep. I'm going to not put my name on that one, though, because there might be some really other important things that are happening. But the cleaning process is really, removing waste, which isn't necessarily built up in AI. So instead, that N2 stage and REM sleep might be the really important things. And that's the building things into schema, that happens. When we're first born, I'm going to try and answer a development question at the same time. When we're first born, and even prenatally, the state of that we're in most is REM sleep. And what state is preserved throughout life is that N2 stage of sleep when we're memory consolidation. When we get older and older, all of our sleep goes down, but that N2 stage sort of remains the same % of our sleep. So, and that during development serves a really important process of building our schema. We need to build schemas in order to be able to know when new information comes, where to file it, right? We need to have a filing system. So when you have a computer and you have no folders yet, and it's just, it could look actually a lot like my screen. If I were to show you my screen right now, there are files everywhere. And when they become too many files, the names start overlaying on each other and there's no organization and you will not be able to find anything. So that N2 stage of sleep, when you're building schema and refining those schema, you need to also eliminate a bunch of stuff. For example, when you learn who your parents are, a duckling learns who its mother is. not only does it learn the features of its mother, but it also prunes away all those features that could have been its mother, but are not its mother. So, for example, anything that's making noise and moving away, it counts as mother. And then it imprints, the duckling imprints on those features. And then there's a huge pruning process going on in the imprinting area of the brain to say, okay, my mother is not red, my mother is not bark, my mother does not, all of these things that could have been mother. But if its own mother is gone and it's imprinting instead on a human who's also, it's there at the right time, it will imprint on all those features. My mother, talks, my mother is tall, my mother is, whatever that human is. And that imprinting is really interesting because it translates to a lot of other things that happens in the duckling. For example, later when it's older and trying to mate, it will try and mate with features that were like its mother, and not exactly its mother, but with other. If its mother was duck, it will try and mate with ducks. If its mother was a human, it will try and mate with humans. So I don't know how I've gone off the topic of AI, but there. Oh yeah, is that Konrad Lorenz? I can't see who it is.
It's Freud.
Oh, Freud.
This is
yes,
Pink Freud. You were talking about thinking what a Freudian duckling.
Right. So anyway, so I think it's also important for AI to be able to, have schema, right? And to know when something new comes in, where to fit that. And I think the last couple of slides that I showed you with that circuit might be the most helpful for figuring out what would be important to implement in AI, because you need to be able to recognize something that you've already learned, for example, and the similarity of the thing that's coming in to something you've already learned, and therefore to put it in the right schema, or to just say, I already know this and I don't need to dedicate more circuits to learning this thing that I already know. So I think those last couple of slides should be most informative. Yeah.
That's fascinating. I think that the kind of garbage collection is close to the cleaning up also a little bit, and also just handling memory in terms of pruning. So pruning happens also in memory networks and memory trees.
Yes.
Deciding what to prune or not matters. So I wonder whether we could create phase-like systems in AI that decides on different types of pruning depending on the kinds of tasks, like a goal-directed kind of pruning. And I think it helps to some extent. As Melanie said, there have been many AI recent papers that use the metaphor sleep, but a very disappointing, in my opinion, trend in AI fields is that they use words from cognitive neuroscience, but they just do something else. And it's very rigorous. It's just more like, it's kind of like a little more catchy than,
Yeah.
They did have something called FearNet that got a little close, as in it kind of simulated different parts of the brain. But it would be really fascinating to have these rhythmic notions of different phases in AI networks. We do in replay situations, there are parts of the algorithm where you experience something in the world and then you go replay things, and then you add something in a priority queue if it's surprising, and then later you replay that some more. So those are some things, but they're not exactly rhythmic in the sense that the brain is. So I wonder at some point whether we could get there. Sorry, this is fascinating. I feel like I'm talking too much. Go back to it.
No, it's fascinating. And the things that you forget, like, for example, I hope all of you will not remember this shirt that I'm wearing. That's not important. Don't tag it. But there are things that we remember forever that aren't important too, right? I mean, that might come up later in a dream and say, I can't believe I remember that. Why do I remember that? That wasn't important to my survival. It wasn't important for anything. But it comes up anyway. And I think it's important for us to be able to, I don't know how to say this, but we don't always know what's important at the time that it occurs, right? It might not be for years before it comes up and we can use it. So,
I find that, part of getting older, to that point you just made, is sometimes you remember a conversation and then you go, oh, now I know what was going on during the conversation. Right, so you remembered it so that you can actually properly understand it only in retrospect. I mean, it's kind of spooky that you go, oh, that's what she meant.
Yeah.
I wasn't really listening, and that retrospect is,
Yeah.
you know, how is one supposed to know that you missed something, so you should remember it so that you get it at some later date, right?
Yeah,
I mean,
great
these things
question.
are also so spooky, right?
Yeah. And again, that early life learning, we don't have conscious recollection of the learning that we're doing, but it is happening. It is, you know, we may never know that the reason why we think of the world the way we think of it is because of these early experiences and the directions that our parents gave to us. But they do formulate our our brains, right? Yeah.
Yeah.
Meanwhile, at the salon, it happens all the time that John disagrees wildly, and then afterwards, like, oh, that's what you meant.
Right. That's exactly what I was trying to signal to you.
Oh, man.
Who else? Mihai's coming in, is that right?
Mihai is what? Oh, I was asking if you would join, but I don't know if he's still there. So I'm just going to read the next question while people decide who wants to join online, and I'll start to bug people. Emma asks, as sleep is very important to survival, if in general we sleep less than the necessary duration necessary for the proper function, is there evidence that lifespan is impaired? So in other words, do people that sleep normally, like maybe 8 hours or such, live more than the ones sleeping less hours per day? Is this small sleep deprivation lethal? And I think not everyone needs 8 hours, so maybe you could also speak to that.
Yeah, no, that's absolutely true. So too much sleep, you can't actually get too much sleep, but those who sleep too much are sleeping too much for a reason. One of the reasons might be that their interleukins, which also induce sleep, which get activated when we're sick, are flooding the brain and therefore making us sleep a lot more. So it might be that those who sleep more than 9 hours a night might need to get looked at for other reasons. You know, are my interleukin levels high? Do I have obstructive sleep apnea, so my sleep is less efficient? Yeah, there's a good reason to sort of look at yourself if you're sleeping deeply for more than 9 hours a night. Those who sleep deprive themselves less than 5 hours a night also die early, but usually because of accidents. Their brain is not clean. Their judgments aren't good. Your forebrain needs that much sleep. Your forebrain, actually, your prefrontal cortex, judgment and decision-making area, is pretty much off throughout sleep. This is actually an area that is pretty much off, except for when it's being cleaned by slow waves. And so we don't know why it needs to be so deactivated throughout sleep, but we do know that sleep deprivation causes impairments in judgment and decision-making, and it's one of the very first symptoms of sleep deprivation. You're less able to control your impulses. If given the choice between salad and cake, you know, whereas you're trying to stay healthy, you might normally in a sleep replete condition choose the salad. In a sleep deprived condition, you will choose cake almost every time. So
I can never turn down a nice bit of cake.
Maybe you don't get enough sleep. I don't turn it down because life is short. But
maybe you need to sleep more, John, then.
Maybe you need more sleep.
A cup of tea and a nice bit of cake. Yeah.
Yeah. I certainly appreciate it. So there are fruit flies which you can breed with, and they're short sleeping, really short sleeping fruit flies. They die fast. And with no sleep, you will die. Every experiment that's been done. Now people say, "Oh, well, I never sleep," but if you bring them in the lab and hook them up with EEG, they sleep. They might not feel like they're sleeping. There might be a tiny area of their brain that is conscious and awake the entire night, but the rest of the brain is doing its job. It's sleeping.
Yeah, or they might be sleeping during meetings, which I miss seeing people fall asleep during meetings in the pandemic.
Oh, yeah. You know, actually, there was a really fascinating paper just released this last week, which tested the hypothesis that if you only let yourself drowse and get into early sleep, you'll be more creative. I would not recommend this for the long term because you do need that brain cleaning and all the other functions. But what they did is they had people holding, I think it was like a metal water bottle can, and they were in a chair, and then they dim the light and let them doze off after doing a test. And it was a serial subtraction task, an insight testing test. And then if they fell asleep, when they fell asleep, they would drop the water bottle, it would clatter to the ground and wake them up. And those that did it this way and did wake up after these water bottle clattering events were 80%, I think it was 80% of the people that did that were able to, when they woke up, gain insight into that task and be able to do it much, much more quickly. Whereas those that continued into deep slow wave sleep, gained insight much less often. And those that didn't sleep at all also didn't gain insight. So I thought that was really interesting. Alexander the Great and several others have done this themselves with, a heavy ball that they hold in their hands. And then when they fall asleep, they drop it. So they get that first dozing state, which nobody's ever studied the dozing state. That's a really fascinating state where there's a lot of synchrony in the brain. It might be that if your need for sleep is really high, for example, in this insight task, you might be able to get into N2 state within that sleep, first couple of minutes before you drop the ball. And so it might be that you're able to gain insights because there is writing going on. Yeah.
What an amazing idea, just so I understand. So if you're trying to solve a problem and you're having trouble, get hold of something heavy, hold on to it.
Let yourself fall asleep.
A little bit in front of the computer screen, drop the ball, literally, right? Yeah. And then go back to the problem and go, aha, that is a great recipe, right?
Yeah, I have never tried it, but it's, that study was a good one. So
I have to post that too. Yeah.
Let me see. It was probably in Science. Let me click on that if I don't make you all go away when I do. Oh boy, you sort of went away. Sleep onset is create a sweet spot. Yeah, that's it. It's just published last week, right? So I'm looking at it. This 8th of December. Yep, that's it.
There's a couple of other questions, so I'm just going to ask them gradually. The next one is that what I understand is that mycelial and fungal network also sleep, especially if spores are not finding the right hosts. They sleep until some proper host is found by movement or whatnot. And that's really fascinating to me because a lot of roots are basically, sharing carbon and sharing resources sometimes happens through mycelial networks. And their sleep and wake cycles could really impact ecosystems. So I wonder, what do we know about the kinds of sleep in creatures and sort of organisms that are not plant and not human, the mycelia, for instance, they're in between, and that their sleep and their entire life is kind of symbiotic with other creatures and they influence ecosystems. What do we know about the function of that?
Yeah, there's a really great researcher named Vladimir Vyazovsky, and he has actually studied fungi and learning. And so fungi can learn. And I don't know myself about sleep, but he was studying the connection between sleep and learning in fungi. So you should look up his papers. They're fascinating. He's a great thinker. Trying to think where he is. He's in England, Oxford, I believe, or Cambridge. I'm sorry for all those
who add his name
hate the fact that I get them mixed up. Yeah.
But what does that mean for
it's like
fungi to sleep?
Because they can't.
No, I mean, they don't have a nervous system as such, right? I mean, what is it even, what are you defining as sleep in that context?
Again, I don't really know, but, let me see. What did they do? Yeah, that's a good question for Vladimir. We had a long conversation about it, but it was many years ago, and I forget now exactly what he meant by sleep in the fungi. But there was, you can't define it the same way you do in animals, of course. But there was some evidence.
Right. So this was a question that I think came up in a few different questions. So as Ida mentioned, in machine learning, people often use sleep and dreaming and so on as kind of a metaphor, right? Because our machines are not literally sleeping. But I'm wondering, in how many of these animal cases or kind of simple creature cases, is it more of a metaphor than really an analogous phenomenon to what we humans do?
Well, so one of the things that defines us as humans when we say we're asleep, we're unconscious, right? So many people don't believe that flies can be conscious. I think that they could be, but it depends on how you define consciousness, right? And the other kind of major thing that we get out of sleep, one of the joys of sleep, is dreaming, the fun of dreams. And although the dream state of sleep in every animal that I have seen does seem to occur in terms of, at least there is another state of sleep. We don't know if these animals are dreaming because we can't ask them that. But from those that have REM sleep without atonia, there's evidence that they were probably dreaming, because they will run and play and swat and hide and all that sort of thing in their non-atonic state. But again, we still don't know if they're dreaming. Now, babies, who we also have a difficulty saying whether they're truly conscious or not, because we can't talk to them, very young babies still have REM sleep. And when they're very first born, there's a ton of REM sleep. So what on earth could they be dreaming about? They have so little experience. Why are they smiling and frowning and kicking and punching? I mean, is this, some people said, is this a past life they're dreaming about? Is it a past life that we forget as we grow? I don't think that's the case, but could be. So instead, there's an interesting group from Iowa, Mark, oh boy. Anyway, I'll remember his last name in a minute. Bloomberg, thank you, Mark Bloomberg, who believes that REM sleep state, the dreaming is just, what's the word, a byproduct of the fact that our brainstem is so active and sending those Pgo waves up to the forebrain, causing us to have all kinds of dreams. But what it also does, that big activity in REM sleep, is it activates our muscles, it activates our spinal cord, it helps us develop our motor systems. And so, and I think that's the case of REM sleep, even when we're dreaming, is it's helping us to make new connections, to prune other ones that aren't important. That's really, really important during development, but it's important all our lives long. So yeah, so I think that those functions are true even of the zebrafish, larval zebrafish. There you see that same big activation in what is probably REM sleep. Another function of sleep I didn't even mention, but to me is fascinating new field, is epigenetics. So the chromatin can be remodeled and is really strongly active and moving around in sleep when it's not moving around in waking. So that chromatin remodeling, the epigenetic control of our genes and expression of different genes probably happens more in sleep than any other state. Yeah.
I just wanted to say in Entangled Life, which is how fungi make our worlds, change our minds, and shape our futures. This is a very, very fascinating book. I'm really enjoying it, with also very beautiful figures in the middle of the book.
Lovely. Right? I'm going to have to read that.
Networks of, whatnot. So Melanie, something I found interesting is that there were some studies that, unfortunately, I don't know why there's not more work on this, and maybe I'll start doing that, where they noticed that they could measure something that's akin to maybe action potentials or the like from mycelia. And so I would love to see, honestly, I would love to learn more about that, especially because they're not centralized, but they do seem to learn things together. They definitely have communication in their networks. And one of the things that this book, and I noticed mycologists in general say, is don't compare forests with brains, because why do you have this mammal-centric idea that the fungal network has to be like, one entity, or that's your idea of intelligence rather than at the collective level, it being in this one entity. So as you know,
I think even brains are a collective. you know.
I fully agree. That being said, we are entities, each of us carrying that collective and then connecting to each other. So I'm very fascinated by collective memory. I've studied it, modeled it. So for me, it was fascinating to think not as individuals about humans, but as just like these connections of networks that make certain things happen or not happen, which are optimal or suboptimal for some goal or other. Likewise, it seems like fungi do that. And something I learned that was very fascinating was trees and plants, they have to kind of bond with the fungi and the mycelial network of the land they are in. And some trees are kind of, they're capable of bonding with multiple species of fungi and some only with one or two. And that determines how this tree migrates and survives after migration to a new place. Also, if
wait a minute. A tree migrates?
Humans obviously take the seeds of trees.
Okay.
Like in the United States, you have a lot of trees that come
from. What did I miss?
Yeah. And there are some interesting ideas that, especially women putting things in their hair, like moving around, or animals just like with fair, just pours something from a tree falling on them, and then they would just go somewhere else, or a bird or such. There are many ways for migration of plants. Which is always in collaboration with other species. But the interesting part was that they need to bond with the mycelia of the new land, and otherwise they can't survive. And it was interesting, if they bond there, one generation later, you take them to the old place, they might not work there anymore. So that was a very interesting thing that I read. So I wonder, with this close connection to plant and animal species, I do wonder whether they have a circadian rhythm, they have a sleep notion too, given they're always underground, like most of them are underground. Like, anyway, fascinating stuff I'd love to learn about. So I'm going to follow up on the work of Vladimir Vygotsky that you mentioned, see what they've done.
So one thing that we were talking about is emergent properties, and that is, it might be that sleep is an emergent property of a conglomeration of communicating entities. So entities being neurons and glia, the whole thing. So it might be that you just can't, if you've got a group of communicating things, sleep is just part of that. It emerges from that. And then someone put in the chat about dreaming of flying, and that reminded me of this book called, I always forget the name, and I don't know if I have it here in my bookcase, but I think it's called When Brains Dream, or something like that, When Brains Dream. And it's written by Antonio Zadra and Robert Stickgold, and it was also just released maybe 1 year ago. And it promotes a hypothesis that's also an emergent hypothesis called the next hypothesis, which means that what we're doing when we're dreaming is we are using the sort of random activity of our dreams to test new hypotheses and to try new things that we can't necessarily do right now in wakefulness, or challenges that we were challenged with that we haven't solved yet. We can sort of come up with, without the judgment of our prefrontal cortex saying that's not possible. It allows us to make new connections and try them out. And when they make sense, to write them into long-term memory, and then learn from these dreams in a way that we can't do when we're awake and our logic systems are all forcing thoughts in a certain particular way. So I don't know if any of you had flying dreams, but they are amazing.
My grandfather taught me in my dreams. My grandfather was passed away at the time. Taught me how to control my flight, because initially the experiences were that I felt like the wind carries me, and then gradually I gained motor control. And I felt confident. It was like, I can fly, people.
And in your dream, it was your grandpa that was teaching you. Oh, yeah, that's amazing.
He passed away when I was a child, so that was like a interesting.
That is beautiful. That is beautiful.
I've also dreamt of living underwater, like an entire city that was underwater. It was like very normal.
Yeah, and so you can breathe and not worry about it. Yeah, totally. I have too.
Another thing, another question we have here is by William, and he says, it is interesting that the harm done by lack of sleep is so severe that people potentially could pass away. But it seems like the hypothesized purposes of sleep, like consolidation and etc., do not seem as physically essential as eating or drinking. Do you have any thoughts on this?
It is as essential as eating or drinking. You actually will die within, I think the longest is 5 weeks if you don't sleep. So it is as essential as eating and drinking, yes.
Wow, I know of people who went on, hunger strikes longer. So.
Yeah. I mean, that's the other thing is you can't store up sleep like you store up fat. Right?
Yeah.
So if you anticipate going on a hunger strike, you can sort of load up on food for over a year and get very enormously fat, and then you could do a hunger strike for quite a long time. But yeah, you can't do that with sleep, unfortunately.
So Habiba had asked about how the sleep process differs in infants, but I think you answered that they're mostly in REM state. Emma asked, is it known if protein synthesis as well as protein degradation are happening in the same level in the wake and sleep states? Do we have more protein degradation when sleeping?
We definitely have more protein synthesis when sleeping. Protein synthesis during waking and also REM sleep is very, very minimal. So it's really during non-REM sleep, that's the time when our chromatin is also really active and transcription and translation is happening really, really fast. And it might be, again, that's harkening back to circadian rhythms, it might be that's the time when all these various compartments have allowed the enzymes and amino acids to be where they need to be all together. Yeah.
I think I would love to hear a little bit of, so I have some work on multi-scale representations in the hippocampus and prefrontal cortex. So I'm very interested in the fact that Melanie mentioned you have a paper on how sleep time scales with brain size. I'd love to hear about that, because also brain size scales with social network size in humans, in mammals, in kind of many species that have brains. So I would love to hear more about that.
Oh, yeah. That paper was really a lot of fun to work on because there was this really sharp transition during development of the amount of REM sleep and what REM sleep is related to versus non-REM sleep. And it was about 2.5 years. And we do know already in human development that the first 3 years are really critical for that whole schema building thing. There's a lot going on. And synaptic pruning is really, really critical. It's not like we don't get REM sleep after 2.5 years, and it's not like nothing's developing after 2.5 years. It's just that it's happening in so fast and so readily in those first couple of years. And that's when REM sleep seems to be the most critical. It's really important actually to recognize this as a parent of an infant, because a lot of parents might think, oh, this is really an important learning time, so I better maximize their amount of time that they spend learning something, flashcards and, TV and, soccer, but actually they really, really need sleep to consolidate that. So do not, my highest recommendation, never wake a sleeping baby and never keep a baby from its nap as well as from its nice solid sleep at night.
Gina, can I ask you a question about the authors? So I see you wrote that with a paper with a bunch of friends of mine, Jeffrey West and Van Savage. So was that looks like a Santa Fe Institute little club. Did you visit there at all?
I did. Yes.
Oh, did you meet Melanie? Have you guys met? No,
I don't think we met. No, but it was a really wonderful place. Wow. And I hope I get to go back.
Oh, okay. So Jeffrey's quite the character, isn't he?
Yeah, I love him. Yeah, he's great.
So Melanie, we're going to have to make sure we're there next time when Gina visits.
I'll let you know. I will
me
let
too.
You know.
Me too, please. Invite me at the same time.
Yeah.
Okay.
Yeah.
Yeah. What a great place, a special place.
Yeah, it was. I also really loved when I visited there. I also really loved Meow Wolf. That became like such an important place for me to visit. And I got
so there are plans now to fuse them, Ida.
Oh, wow.
What?
I'm joking.
Oh,
Hey guys, my computer's
I
on
was gonna get my tickets now.
my computer's on 9% battery, so I can go get a charger.
Okay,
but
yeah, we will wait for you.
Okay, I'll be right back.
I don't know. So John and Melanie, I was so serious when I was trying to solve the puzzles and look around. Some people thought I'm a part of the museum and started following me around. They thought I am playing a scientist in the museum. Like I'm a performance artist performing a scientist who's trying to solve the problems. It was fantastic.
Were you sort of overly frowning and overly going, and things like that? You were like this, you were going,
That's your facial expression. No, I was wearing like all black, and then I had bought like a cap that said Meow Wolf, that like very subtly. And then I was just going around and figuring things out, like reading the books that was there, looking, testing things, and then all of a sudden opening a door that people didn't see. So I was really playing around to find that space, and people thought I'm a part of it. At some point, some people start following me. I was like, oh, I'm making new friends.
well, there's the danger of that with me, Ida. My response to the puzzle was, oh, whatever.
Well, if your Microsoft career doesn't work out, you know you have other job possibilities.
Absolutely. I mean, I painted that thing and all of these. So yeah, I'm definitely going to go to the art work. I was
going to tell you, Ida, that painting something I'm going to write into my long-term memory. I know I'm going to dream about it later.
I'm so happy you say that, because it's me and my dad and my sister, and I will send you a JPEG of it so you can.
Oh, that would be great. Hey, you should make it what is it when you make your art? There's one copy that has all the special things, and you can buy art that way. Yeah, whatever.
I know what you mean. We just had a talk about this at Microsoft yesterday. I think for me, art is a way of tuning out, and I feel like all of these things is way too much tuning in, and I'm like, no, my job is tuned in already.
Yeah.
Did you mean an NFT, or did you mean like what is it, ledger? I forget what the name is, like the digital certificate that you have.
Yeah, it's a digital certificate. Yeah, so you can actually sell art digitally. Yeah.
All right. So our next question, and we had more questions added, so I'm going to try to go faster a little bit. We have which features of sleep do we see the most overlap with periods of awake, eyes closed rest? To what extent can portions of sleep be replaced with eyes closed rest?
Yeah, I don't think it can be replaced by eyes closed rest, because there's a very particular neurotransmitter profile of sleep that, for example, slow wave sleep, the acetylcholine systems are off. There's no acetylcholine in the forebrain. And in fact, animals that sleep unihemispherically have no acetylcholine in the hemisphere that's sleeping, and a lot of acetylcholine in the hemisphere that's awake. So when we're awake, we have acetylcholine, and the cholinergic system is on. It's kind of a definition of the system. REM sleep is called paradoxical sleep because it is the most like waking in a lot of ways. So we have tons of acetylcholine during REM sleep, lots of neuroplasticity, lots of activity, desynchronized activity, different areas active at different times. But even that is completely unique from waking, in that the noradrenergic system of the Locus Cyrillus is off normally and optimally, and the serotonergic system of the dorsal raphe system is off. And those two systems are on throughout waking. So eyes closed waking wouldn't be able to serve the synaptic bidirectional plasticity that REM sleep serves. However, there is another state which we haven't looked into because we can't ask animals to do it, and that's Transcendental Meditation. So that state actually has a lot of theta activity like we have in REM sleep. What we don't know is, because we can't do it to humans, is what's happening to these brainstem nuclei, what's happening to the Locus Cyrillus. Does it look like REM sleep too? Does the serotonergic system look like REM sleep as well? But there are claims that people in transcendental meditation can do that to replace sleep. It's just not well studied on a neurochemical level, even an electrophysiological level. And no one has done a long-term study to see if people who are in that transcendental meditative state are not sleeping sometimes. You know, we just don't know. Yeah.
That's very interesting. The next question is by Samantha, who asks, which features of... Oh, sorry. I did already read that. The next question is by Mihai, who asks, why do sleep-related processes you mentioned happen during sleep? Could processes such as synaptic pruning, memory consolidation happen while awake instead? So why is it that, and maybe you answered already, it's because of the cholinergic system, but maybe you want to mention.
Yeah. Yeah. So the cholinergic system needs to be on when we're learning something new. The immediate early gene system is on also during REM sleep as well as waking. But again, with the synaptic pruning, you can't do it when norepinephrine is present, and norepinephrine is present throughout wakefulness. So there are certain unique functions that just can't be done during wakefulness. Yeah.
Why is it like, why did this happen like that? Are there any species that don't, are there any species of mammals that don't have sleep, or what are the species that don't have sleep?
Okay, so it was thought that the platypus doesn't have REM sleep, but in fact, if you look into their brainstem, almost all their sleep is REM sleep. So there's some studies by Jerry Siegel, who does studies of animals all over the world, and including in zoos. And what he's found is that elephants in captivity, once they've had their calf, don't seem to sleep much. At least the calf doesn't seem to sleep much in the first month. It's running around almost all the time, and the mother doesn't sleep much because the calf is running around all the time. And that's also true of whales. You know, when killer whales they studied in captivity, when they calf, they also don't seem to be sleeping much. The calves need to breathe more often than the adults do, so they have to go to the surface and breathe. But no one has ever done any depth electrode studies of these animals. So while they're moving, even walking, they still could be sleeping. We know we can sleepwalk, right? So we need to dig deeper, I think, and do better studies to see if there are long periods of time. It used to be thought that seagulls, not seagulls, but I think they're big cormorants, big sea-going birds, would be out there for long weeks, and they're flying the whole time. They don't dip their wings into the water. They're not waterproof. So they're out there for long periods of time flying. So it's thought, well, maybe sleep isn't important for them for these weeks. But in fact, when Niels, can't remember his last name right now. Anyway, he's the person who studies birds and sleep. When he outfitted them, he found that they are sleeping even while they're flying. They soar, and they're sleeping while they soar. And then one hemisphere wakes up and they turn, and then they continue to soar. And then the other hemisphere wakes up and they turn, and they continue to soar. So they start really high and they circle down.
So this notion of hemispheres, one hemisphere being in one sleep cycle and the other one, and being quite partitioned, is that true? In other words, is it really that you can have almost independent hemispheric cycling through sleep? I've always wondered if that's really true.
Yeah, it is. It's really true. It's really a beautiful thing, and it puzzled sleep researchers who thought there had to be central brainstem, sleep generators, and that seemed like they bilaterally project. So how could you have one hemisphere sleeping at a time? But it's just organized in those animals. We don't seem to sleep one hemisphere at a time, but those animals where it's survivally important, it's important for their survival, they definitely do. So there must be just a slightly more unilateral organization to their brain than ours is.
So do you think you mean that it may bifurcate even down to the level of the brainstem?
Yes.
I see.
Yes. Yeah.
How interesting. I've learned about 100 fascinomas today. Right? All right, okay.
It is fascinomas. I love that word.
Yeah. It makes you wonder if, in humans who've had their corpus callosum severed, sort of what does sleep
That's a
look
great
like?
question. I don't know that anyone's ever studied it, not that I know of. So
that is awesome.
Yeah.
Wait, do they still sever the corpus callosum, or have they found other methods too? Yeah, okay.
I think they still do. Yeah.
Okay.
Yeah.
I would love to see that study, actually.
Yeah, me too. John says there's a lab that studied a human with one hemisphere. They sleep fine. Yeah, and in fact, you can sleep fine even without any cortex at all. There are still sleep cycles that our brainstem generates, so sleep-waking cycles.
Yeah, but if they're severed and they're separate, that's very interesting. If they both exist, both cortices, but they're not connected.
They're not connected.
Actually, it's interesting because I wonder about sleep and mental illness, because I know that one of the diagnostic criteria of bipolar is that they in the manic phase they just don't sleep, and in the depressive phase they sleep a lot. And then I know that in schizophrenia it seems like there is a rapid rate between the two hemispheres, just like if you take an EEG of them, they switch more rapidly, which is why they the kind of optical some optical illusions they don't experience, or some multi-stable stimuli they can see faster, because it switches too fast for them to pause on one or the other. So it seems like there are some rhythmic and sleep related, and also we know about issues with sleep in PTSD. So and in also in depression. So it all of a sudden increases or decreases. What is the kind of, it's not just serotonin, right? So what is the reason for all of the relationships between, and which way is it? Is it that some people say that, if you fix the sleep, you fix so many of the different mental illnesses, because some of them is just related to that. So I wonder, what do you think? Which way is it?
We really don't know. And the thing about sleep is that there's redundant systems, thankfully, because sleep is so important. There are many different areas in the brainstem and even in the basal form, not the basal form brain, the hypothalamus that regulates sleep, and the thalamus. So if one system fails, another system can still take over. And so I would love to see tests is whether you can fix the sleep and therefore fix the mental illness. But with schizophrenia, for example, the lack of sleep spindles is really the most startling electrophysiological signal of schizophrenia. So
I actually didn't know that.
Yeah, it's interesting, isn't it? So no sleep spindles, or very small and very, very weak. So if sleep spindles are so important for schema building and cortical connectivity, and so it would make sense. So there was a study that tried to fix sleep spindles with a sleeping pill that preserves or increases sleep spindles. They were able to increase sleep spindles a little bit, and they were also able to help. It was a small study, I think 27 people with schizophrenia. They were able to help them learn from sleep. It would be great to do a long-term study to see if you can, give them if they're able to take these over a period of a year, whether you could start to rebuild some of the schema and sort some truth from falsehood kind of things. No one's ever done that study, but it's also not perfectly preserved their sleep spindles by this sleeping pill. So maybe there could be another way to restore sleep spindles in those with schizophrenia and make them more normal and do what they're supposed to do. But yeah.
And
Gina, sorry. No, I was just thinking, listening to you, they're like, sleep seems like to be one of those meta subjects, right? It touches on everything, right? I mean, and the three sort of areas, I always thought, movement was a meta subject, exercise, right? And the three areas that really matter for people's health, right? Nutrition, sleep, exercise, and movement.
Yeah.
Right. And it seems like you can almost, in any one of those meta subjects, cover almost all of life. Is one of the interests for you, I was just, I hope this isn't too personal a question, but is one of the interests in sleep for you, is it because it's just so protean in what it touches upon? I mean, it's just so, we've been talking about mental illness, we've been talking about fungi, and there doesn't seem to be anything where do you think it's just like the meta-rich subject, compared to...
It is, but it's something that I've only discovered as I go along, because as I find found out more and more about sleep. And that might be the reason that I've stayed in the field, because it's never boring. There's always something new, and there's so much that we don't know about it. I totally agree with you. I got into sleep because I went to a conference, a very small conference, amassing almost all the sleep experts in the world at that time in 1988. It was a small conference, and among all of these experts, no one had a clue. I mean, everybody had a hypothesis, a favorite hypothesis. Why do we sleep? All of them seemed very niche in terms of, we have REM sleep with rapid eye movements because we need to move our eyes so the vitreous fluid, it doesn't get stagnant. Well, a lot of animals don't have eyes, that can't be the function of REM sleep. But, and anyway, so... But there were really some fascinating hypotheses that just there weren't enough people in the world to test. So I realized, as an individual just freshly graduated from undergraduate, that this is a field that, I can come up with some harebrained idea and find out ways to test it and actually, give to the field in some way. And so, science is long and life is short. I don't believe we've figured out why we sleep, but I think we figured out a lot of reasons why we sleep that seem to be really important.
And then the final thing, when you mentioned, how sleep, people who say they don't sleep, and I don't know if this book, I'll make a book around that. Did you read that book called 24/7, Late Capitalism and the End of Sleep?
Yeah,
brilliant
I haven't
essay
read it,
book.
But...
I think you'd love it, by Jonathan Crary, Crary. And what was wonderful about this book is he opens by saying that the capitalists don't know how to make money out of the period when you're asleep, right?
We can figure that out.
So they basically thought, what about if we get rid of the nighttime? So there was this idea that you put these rows of reflecting satellites, so there would be sunshine on the dark side of the earth, right? So you could hijack sleep. So it's just horrifying, right?
Yes.
So I just think that there's something about sleep being the last bastion of escape, right? And I think you'd love this book. It's quite a interesting meditation.
I think I've seen it, and I think Vlad Vyazovsky has a chapter in there. He has an essay in there. So you might be able to find his spelling of his name through that.
And it's such a great title, right? Late Capitalism and the End of Sleep. Oh my God.
Yeah, very great title.
All right.
So first of all, I found Vlad's website. I think it's that one from Oxford in the chat. So check if this is
SKIY.
The other thing I wanted to say is I think maybe, so first of all, John, like one of the advertisements that's, when you are in the tech world, you constantly receive these advertisements for how to manage your stocks and what stocks to buy. And they're like, make your money work for you while you're asleep. So they suggest that if you buy that's their tagline, that hey, you can go to sleep, and in order for that to happen, that you can make money during your sleep, come and buy these stocks or work with our advisors. Another thing, it was this guy, I don't remember what his name was, that he was saying, oh, just tune out and everything. So he was promoting like totally like just going back into a state of like only coming to surface sometimes and staying in this kind of intuitive, dreamy state, this kind of to be a disruptor in tech. So there is that subculture in tech too.
What a horror.
I know.
Well, Gina, it's midnight for me, and I have to get up at 4 a.m. to fly tomorrow.
Dear.
But, so I'm going to get some sleep.
Yeah, I hope you
have the ultimate excuse. You can't criticize me.
No, I do not. And I don't
but I just
criticize
want to
my
say,
students
on
when they
behalf
fall
of the
asleep
Salon,
in class too.
I don't think it's been this much fun. It's just wonderful, and you're just a phenomenon yourself. Thank you so much for sharing all this stuff with us. And
so much more to talk about. Thank you so much.
I know.
It was great fun.
All right.
We need more things with you. We need more events with you.
Yeah.
I'm
night,
willing.
Melanie. Night, Ida. Night, Gina.
Good night, John.
Bye.
Good night,
Bye.
Sleep well.
I'm going to be thinking my Locus Cyrillus is too hyped up by this conversation.
Yeah. Could be.
Bye.
Bye.
Thank you, Gina. I don't know what your time limits are. I don't want to hold you.
Well, I'm supposed to have a lab meeting in 15 minutes, so I might have to get there.
Then let's try to ask maybe one more question or two or wrap it up. So I think we asked most of the questions, which were, I think, pretty great that you answered all of them. Someone followed up on Melanie's question about older vs younger sleep. And they asked, do we become less able to learn because of reduced plasticity or other reasons? So the brain somehow decides that less lower quality sleep is enough. And or do we get worse sleep and thus we end up learning, retaining rest? And so I would, and then they add, or day by day, are sleep patterns different after days in which we take in a lot of information vs more boring days? In other words, if we keep learning new things and like whatnot, do we even in older age sleep more?
Yeah. The answer is yes. It's in every study done, the density of our sleep spindles and the amount of that sleep spindle state goes up if we're able to learn and consolidate through sleep. So as the sleep, and also our sleep goes up with the sleep when the homeostatic demand for sleep goes up, which includes when we've learned something new that we need to rewrite our brains for. So I also want to just mention, since this, in answer to this question, it's sort of indirect answer, sleep is also disturbed in drug addiction. So any drug addiction, sleep is disturbed, and disturbance of sleep, people reporting they get less good sleep, more insomnia, precedes the relapse into drug-seeking behaviors in those that have been rehabilitated. So again, if you can fix the sleep, and it might be because, withdrawal of drug is very stressful, and our Locus Cyrillus fires and says, "I need it, give me that drug again," and then wakes us up and keeps us from sleeping. That could be a positive feedback loop that then causes us to seek drugs again. So I say us, not, thankfully I don't have it, but those who do, and it's not a judgment. It's really a physiological dependence that's that needs to be addressed. So I think sleep needs to be addressed in people who have had drug dependence, and it might be that if you can address the sleep, normalize the Locus Cyrillus activity, reduce the stress of that withdrawal state, you can actually also, in that way, help the hippocampus be restored and refreshed so that we can better learn and remember the negative parts of being on drugs and the positive rewards of being off of it. So it might all be related. This is a small study we started with a new graduate student in my lab that I'm really looking forward to, with animals, looking forward to seeing the answers to that.
That's really exciting. What animals are you using?
Rats. Yeah, rats seem to have the same preferences we do.
And it's different from mice, right?
Yeah, rats are a little different from mice. Yeah.
Very interesting. All right. There are more questions, but I feel like this is a good time to give you a couple of minutes before your lab meeting. This has exceeded. I mean, I already expected all, but this exceeded my expectations.
Oh, that's so nice.
Thank you. This was so fun. Maybe one last personal question. Why do I get grumpy after afternoon naps?
Yeah. Oh, after afternoon nap. I do too. So what I found works really well is a sweet, like chocolate, lollipop, even actually eating anything, but especially if it's sweet, it just wakes me up faster, and the grumpiness melts away. I don't, but I do know that the sympathetic nervous system, which is involved in grumpiness, is calmed by sweetness. So it seems to, even in the mouth, you don't even have to swallow it, but something sweet in the mouth, and even fake sweetener does the same thing, activates the same things. So it somehow feeds back into the brain and calms us down. And maybe you just, had a lot of good things happening during sleep that you need more food for. You need food to restore.
The reason I ask is because in my night sleep, I wake up in the morning usually hyper and happy and like in a kind of a euphoric state. But afternoon sleep, usually I wake up in an existential, like, oh,
Yeah.
way.
Nobody knows why, but I have felt the same way. Yeah, just try and give yourself some chocolate.
Yeah, thank you. I love that. This is the perfect note to end on.
Yeah.
Thank you, everyone. Thank you so much, Melanie. And I really hope that we can have you in other talks maybe next year at MSR. I mean, with the world being closed, probably also virtual. But also, I hope that we get to discuss the topic of sleep more. I really learned a lot, and I want to thank you on behalf of everyone. I thank also our audience, and please send your questions. This has been so much fun.
This has been so much fun.
And happy
Get
New Year,
Vlad.
everyone.
Get Vlad next to him. He'd be great.
I was exactly thinking, I was like, I should immediately write an email.
Yeah.
Yeah, I've learned a lot about sleep because of James Antoni, who's one of my best friends, and he works with EEG of sleep, and he's building models of it now with Randy O'Reilly. But this was like so much that I had not learned about from all of these events. So I really thank you.
All right. Thank you. Really nice to meet you.
Have a good one. Bye.
All right.
Happy
Thank
New
you,
Year, everyone.
Happy New Year.